Okay. Well, Ella, what is this? It's cellular respiration! Woohoo! So, Ella, what is cellular respiration? Well, Naomi, cellular respiration is the formation of energy in the body. It's the breakdown of glucose to form ATP, water, and carbon dioxide. So, the breakdown of glucose is used to synthesize ATP from ADP and inorganic phosphate. In the cell, many reactions occur. These reactions take place in the cytoplasm and the mitochondria, and are exergonic reactions that release free energy. In the reactions, oxygen is reduced to water, and glucose is oxidized to form carbon dioxide. In aerobic cell respiration, there are 20 reactions that occur in the presence of oxygen. The 12 hydrogen atoms break away from the glucose when it gets oxidized, and carry electrons to oxygen to form water. The remaining oxygens combine with the carbons to form carbon dioxide. In anaerobic respiration, cells can oxidize food molecules without oxygen, using either regular anaerobic respiration or fermentation. These only release small amounts of energy, though, compared to aerobic respiration. There are two types of anaerobes. Obligative anaerobes have to live in an oxygen-free environment, and facultative anaerobes can live in an oxygen-free environment or in the presence of oxygen.
Okay, let's get started. So, for the first step, we have glycolysis. Now, it doesn't actually occur in the mitochondria. It occurs in this area here, which would be the cytoplasm. Okay, so first we start off with this glucose molecule. Okay, so for our first step, 2 phosphate groups, which would be those there, go onto our glucose molecule. And it's transferred there by the phosphorregulation, where ATP is converted into ADP. This will result in a fructose 1,6-biphosphate. Okay, so here we have a picture of the fructose 1,6-biphosphate. As you can see, it has the 2 phosphate groups on its end. So this first step in total, the process, is called glucose activation. Okay, so now our second step is the sugar splitting. So the fructose 1,6-biphosphate is split into 2 separate fragments called G3P and DHAP. The DHAP is then converted into the 3GP. Therefore, this product makes 2 molecules of G3P. So here are the G3P molecules. Okay, so now we're on our third step, which is the oxidation. The 2 G3P molecules become oxidized using NAD+. The NAD+ is turned into NADH. This releases energy. The energy released is used to attach phosphates to the sugars, making them 1,3-BPG. So this illustration now shows that the G3P molecules, which would be 2 in total, are being converted down to the 1 to 3 BPG, and this releases energy and creates 2 ATP. Okay, so now we're at our last step for glycolysis, which would be the formation of ATP. So in this step, the phosphate groups of the molecules are transferred to ADP. This creates ATP. This occurs due to the process of substrate-level phosphorylation. Therefore, the end result of the glycolytic pathway is 2 ATP by substrate-level phosphorylation. Okay, so now we're in stage 2 of the entire process, which is pyruvate oxidation. So pyruvate oxidation actually occurs in the matrix, which would be this area here. Okay, so once the pyruvate molecule has entered the matrix of the mitochondrion by the membrane carrier, the coenzyme A is attached to the acetyl group of the pyruvate molecule. This enables it to be oxidized. Furthermore, the NAD reduces down to NADH, which is shown right here. The remaining carboxyl group is removed as waste, which is the CO2. This produces an acetyl-CoA. The acetyl-CoA will enter the Krebs cycle to continue aerobic respiration once energy is required. In total, this section had produced 2 NADH.
Next up is Krebs Cycle. Now, this cycle is a third stage in cellular respiration, and it occurs right in here in the matrix of the mitochondria. Whoop-bam! So, this cycle has 8 reactions that transform energy into usable forms, like ATP, NADH, and FADH2. These reactions release CO2 as waste. For every glucose molecule that enters glycolysis, Krebs cycle goes through 2 whole cycles. Holy shimoly. So, step 1 in the Krebs cycle. So, first off, acetyl-CoA reacts with oxaloacetate to form citrate, and the CoA is removed. In step 2, citrate goes through isomerization to form isocitrate. In step 3, isocitrate is oxidized and becomes alpha-ketoglutarate. An NAD plus ion from the matrix is produced. It gains electrons to form NADH. And then, in step 4, alpha-ketoglutarate binds with NADH and is oxidized by NAD plus to form succinyl-CoA. CO2 is removed. So in step 5, right here, succinyl-CoA, it loses its CoA and becomes succinate. A phosphate from the matrix displaces the CoA, and GDP is phosphorylated to become GTP. The phosphate group is transferred to ADP to form ATP. So you got one of those ATPs right there. Look at that. There we go. And then in step 6, right here, FAD comes from the matrix and oxidizes succinate to form FAD2. Then the succinate becomes fumarate. And then in step 7, right here, H2O is added to the fumarate to form malate. Super simple. And then number 8, malate undergoes a redox reaction with NAD plus to make NADH and oxaloacetate. So this cycle repeats 2 times if we remember. So once it gets to the 8th reaction that isn't numbered on here, it just starts the whole entire cycle over again. So basically, in summary, there are 6 NADH formed, 2 ATP formed, and 2 FADH2 formed for each glucose molecule that enters glycolysis. So that's after 2 cycles of the Krebs cycle. Next, we have the electron transport chain. The electron transport chain, ETC, creates a lot of ATP through oxidative phosphorylation. So ETC is found in the inner mitochondrial membrane, all up in here. Okay, so in the ETC, there are 7 main components. Out of those components, there are 4 complexes, oxygen, and 2 mobile electron carriers. So complex 1, right here, is NADH dehydrogenase. And complex 2, right here, this pear-shaped nugget, is succinate dehydrogenase. And then complex 3 is cytochrome BC1. And complex 4 is cytochrome oxidase. And then you have oxygen. And then the mobile electron carriers are ubiquinone, which is just like UQ, and cytochrome, which is just Cyt C if you want to do like a little short form. Bam. So, first off, the proteins in the ETC are lined up in order of increasing electronegativity. So like everything's like in order there. So from the bottom is the least in electronegativity, and over here has the highest. Okay, so first off, electrons or energy is passed along each component through the membrane. Okay, so NADH goes through the NADH dehydrogenase, and FADH2 goes through the succinate dehydrogenase. Next, hydrogen ions are pumped into the intermembrane, and UQ carries electrons from NADH dehydrogenase to cytochrome BC1. So, that's an arrow. And then, Cyt C carries electrons from cytochrome BC1 to cytochrome oxidase. Let's see. So it'll go right there. Look at all these arrows. They're so beautiful. Okay, and then FADH2 starts at the succinate complex, so it's like has a shorter kind of thing than everything else. So it starts, instead of starting here, it starts here. So in total, NADH produces 3 ATP and 3 hydrogen, and FADH2 produces 2 ATP and 2 hydrogen. So that's pretty good. And then when the 2 electrons pass through cytochrome oxidase, the very last one, they combine with oxygen and hydrogen, which make H2O. Oh yeah. So H2O is the only element with a high enough electronegativity to actually strip the electrons from the last component. So that's why it's used there. So overall, the goal of electron transport chain is to create an electrochemical gradient in order to power the synthesis of ATP, and then altogether, by the end of all of this, you'll have a whole whack-a-mole of ATP.
Finally, we have chemiosmosis, also known as the proton pump. Chemiosmosis only occurs after electrochemical gradient is created by the ETC. ATP synthase, embedded in the inner membrane, forces the H ions through, so that it flows from the intermembrane space into the matrix. H protons provide energy from their movement, which provides a bond between the ADP and inorganic phosphate. This is how the ATP is made, by oxidative phosphorylation. So, we heard that you heard about our wonderful project and watched it, so now we have a wrap just for you. Yo, Ella, did you know? Aerobic respiration is broken down into 4. Step 1, it's fine. Glycolysis is number 1. Occurring in the cytoplasm, loving that glucose and adding them phosphates, and it'll be like new, yo. Producing pyruvate, isn't that so great? And no worries, it gets rid of that H2O waste, making 2 ATP for, yay, you and me.
Oh, Naomi, my homie, I know all about that pyruvate. Pyruvate is great. It's stage pyruvate oxidation. Coenzymes NDH make it hard to rhyme, but its products are define. Acetyl-CoA pushes it forward in the stages A, but no ATP is displayed for this stage.
Krebs, oh Krebs, we know about that respiration stage. It's chilling in the matrix, producing oxalacetate. What? 6 NADH and FADH2 working together to form that ATP times 2. Oh, and throw out that CO2.
Now let's go ETC, hiding in that inner mitochondrial membrane party, making ATP like a boss producing 34 ATP with only a little water loss. So now you know aerobic respiration happens in 4. Glycolysis, pyruvate, Krebs, and ETC together make an amazing cell-breathing party.
Step 1, Step 2, Step 3, Step 4, cellular respiration. Here we go. Word.
Okay, what class do I have? Oh shit, I have
math. Have not been saved. Do you want? Yes. Save.